Translational research helps bridge the gap between what we're learning in the laboratory and what makes a difference in the lives of human beings. And our first speaker is Dr. Kishore Guda, who is a veterinarian and a PhD, who is at Case Western. And he is looking at the mutational signatures of African American colorectal cancer, and also just doing some very interesting work. We look forward to hearing from him, Dr. Guda.
Thanks, Susan, and really
Good morning.
Honored and humbled to be part of this group. And I hope that you're seeing my screen. I was listening to this, and Taelor Scott, as someone who lost his dad, when I was a teenager, at least I can understand the vacuum that it creates, and he passed away due to a disease, and this is also really personal to me, and can we do something about it? So today I'm just gonna talk briefly about some of the work we have been doing to understanding the colorectal cancers, particularly in African Americans, more from a molecular angle. This is a recent statistic from American Cancer Society. The number of folks that will be diagnosed with colorectal cancer, and a third of them would likely die from the disease. But more importantly, what we have seen and known is that the African Americans still continue to exhibit the highest colorectal cancer incidence as well as mortality rates. So there seems to be a clear unequal colorectal cancer burdens in that population. And as shown in this graph, you will see, if you consider the incidence as well as the mortality rate per 100,000 people, and if you focus on these two groups, non-Hispanic whites and non-Hispanic blacks, you can see that the African Americans do have a 25% increase incidence of the colorectal of this disease, both in men and women, as well as an increase in up to 50% mortality rates in both men and women. So clearly, the African American population are disproportionately affected by this cancer. What is also an interesting fact is that typically colorectal cancers in the general population, or if you take Caucasians, you would often see them in the left side of the colon right here. But what is interesting is in African Americans, these cancers tend to be formed on the right side of the colon. So there could be potential genetic or molecular differences or microbiome, as we have heard from the previous scholar speakers, that could be somewhat contributing to this differential anatomic site where you often see these cancers. Of course, as we all know, I think the biggest factor for some of these disparities or the unequal burdens is socioeconomic and access to care factors. One of the things that we are doing at our cancer center is also having these researchers go on listening to tours in inner cities, where we go and talk to these wonderful old ladies and sort of in an informal chat. And what I often hear is, I couldn't get a ride to the hospital. I didn't check at the right time. There are also economic factors preventing them from getting the access at the right time. So I really think there's clearly the majority of these is due to these socioeconomic as well as access to care. But given these differences, what I just alluded to, could there also be molecular differences in these tumors that could potentially contribute to these unequal burdens? So when we asked this question and we looked at the databases, specifically TCGA and other databases, what we found is there is a dearth of, these cancers being represented in the public domain or in the public databases or cancer databases. And we also found they are being understudied, the cancers arising in these ethnic groups. So we therefore, initiated a study to perform some in-depth characterization, at least at the molecular level, specifically on colorectal cancers arising in the African American population. So we used a high throughput genomics approaches, and through our hospital, we are able to accrue or 100 African American as well as Caucasian colorectal cancers as a comparator to study the molecular landscapes. So, and as an initial approach, we're trying to look at what we call as genomic copy number alterations, meaning in cancers, you do often see that some certain regions or some genes, they tend to increase in their copies. It's basically you're copying and pasting the same gene over and over again, that you won't see that in these normal cells. So we initially wanted to profile these, what is the landscape of these copies that you see in the colorectal cancers in African Americans, and using a genome-wide approach. So at that point, we were not happy with the algorithms that are available to profile these things. So we came up with a new algorithm. It took us 6 months, where we developed this program called ENVI that can specifically look at these alterations in a clinical tumor sequencing data sets. And using this algorithm, so we then fed the genomics data from the colorectal cancers and try to look at the alterations. So shown here in this graph on the top are these chromosome numbers, actually, including X and Y. Red indicates a particular region or a gene has gained more copies, whereas blue indicates a particular region or gene that has lost one of the two copies. So the on the y-axis, you will see the frequency, what fraction of the cancers that we are seeing such events. What we found, at least in this graph, this is typical of colorectal cancers. All these regions, the chromosome 7, chromosome 13, loss of chromosome 18 region. These are all highly typical of and well known in colorectal cancers. But however, we also found certain regions of focal alterations, very pinpointed alterations that we were not able to detect or identify in cancers from the Caucasian population or in the public domain. So currently we are actively focusing on these regions to further categorize these things. Then as a next approach, what we found is, in addition to the, or what we wanted to attempt is to, in addition to these copy number alterations, can we also look at the errors or the mutations in certain genes in the cancers, which is a well-known underlying phenomenon, how cancers do develop due to these genetic alterations, or we call the genetic accidents. So I'm not going to go through this entire thing of how we developed this, but the outcome of that is, if you look at the most well-known or well-characterized cancer driver genes, there are 6 or 7 of them, at least in the colorectal cancer space, but many of these genes are as well altered in other types of cancers outside of colon as well. So these are well-known cancer driver genes. So we compared the mutation frequencies of these genes among African American, the Caucasian population, while we did not see much difference, in terms of frequency-wise, what we found is this particular gene, KRAS, which is actually an oncogene, well-known oncogene, and there are also active clinical trials going after, how do we target the RAS oncogene as a potential targeted therapy for colorectal cancer. What was interesting is that we found a higher frequency of RAS gene mutations in African Americans compared to Caucasians. And we have done this study, we have replicated this study, and this effect is not only in our study, but also the studies performed across the nation. There are a couple of studies that were done in African American colorectal cancers, where they have also reported a higher frequency of RAS oncogenic mutations. So in addition to these well-known cancer driver genes, then we asked, are there other genes in the cell? There are roughly like 20 to 30,000 genes. We asked, there are other genes that are showing these differences in frequencies, mutational frequencies, other than the well-known cancer driver genes. In fact, we did find a set of 15 or so genes showing a higher mutational frequency in African American colorectal cancers. For example, mutations in these 15 genes accounted for up to 40%, 41% of African American colorectal cancers, as opposed to only 15% of Caucasian CRCs. Now, once you find this, could there be a clinical significance or association for this difference of mutational frequencies in these 15 genes? It turns out that if you are mutated, or if those tumors have a mutation in any of these 15 genes, those tumors tend to recur at an early time point than those that did not have mutations in these 15 genes. So if you consider both early stage 1 up to 3, as well as stage 3 cancers alone, colorectal cancers alone, we found this clear distinction or demarcation where mutations in these genes, those cancers tend to recur sooner than those that do not have these mutations. One of those 15 genes is known as FA6, and I will tell you why we are interested in this. And we also are thinking this could be a potential novel colorectal cancer driver gene. And this is just showing the protein structure of the FNA6 and these mutations in that gene. But this gene belongs to what we call an ephrin receptor superfamily of tyrosine kinases in the cell that perform various different functions in health and disease. And not only that, 2 of these members related to this ephrin family were previously identified as significant genes that are mutated in Caucasian colorectal cancers. However, until now, the role of this particular gene, remains unknown. So we pursued studies in characterizing the biologic significance or role of this gene in colon and during cancer development. So shown here is a cell. This is the cell membrane on the nucleus. So FNA6 is a receptor tyrosine kinase. It relays the signals from outside into the cell. There are certain changes, biochemical changes that happen when it binds to its preferred ligand, and then it transmits the signal into the cell. It does this by bringing in more partners and then relaying that signal to intermediary partners, and then those go into the nucleus and then bring about changes of certain genes that can express as a particular phenotype, or a function. So since we don't know anything about this, our goal was to understand what the binding partners are, signaling mediators, or what are the downstream targets, and what would be the phenotype. And similarly, we are then studying how the normal receptor can differ from these mutants that we identified in African American colorectal cancers. So using a series of high throughput proteomics, genomics, biochemical assays, what we found was at the receptor level, where you induce a chemical change, we call it the phosphorylation, that is essential for the activity of the receptor. A couple of these mutations that are derived from the colorectal cancers showed complete loss of that activity. We also found differences in the binding partners between the wild type as well as the cancer. We also found alterations in these genes and the downstream involved in inflammation, metabolism, energy balance, etc. So currently we are focusing on how these mutations alter the properties or potentially respond to standard chemoradiation therapies. We also developed mouse models where we knocked out FNA6 or induced a mutation, developed, engineered the mouse models with these mutations. Here is an example. When you delete FA6 in the colon, you would see an altered length of the colon crypt. This is just a microscopic image of the colon, how it looks if you delete the FA6. We also find, interestingly, there is an increased proliferation or the growth rate of these crypts when you delete this particular gene in the colon, and also how some of these mucins, these are just the composition of these mucins that play an important role in normal colon maintenance differ. So currently we are looking at the characterization of these colon mucosa. We are also trying to study the outcome of this from a colon cancer perspective, using a spontaneous or carcinogen treatment or crossing these mice with the APC driver gene model. So the significance or applications is basically what we learned. If you do in-depth studies in tumors, there is a potential to identify or reveal, unravel new cancer genes. If those 15 gene mutations that we found behaves differently or more aggressively, we can actually establish some early testing strategies or treatment strategies to those patients. Or if the chemical mutate, the pattern, what we call the signature of these genetic mutations in these cancers are the result of environmental or dietary carcinogen, then we can instigate epidemiologic studies to potentially identify the source of exposure, providing strategies for chemo prevention. And finally, with the efferent in particular, it is a hot topic under active investigation as a target for cancer therapy. So our studies in the FA6 preclinical models could open new avenues for targeted therapies in cancer patients. So with that, I would like to end. Particularly, this work is being led by two of my postdocs, Sri and Durga, and these are all my collaborators from CASE, Joe Willis, Sandy, and Vinay. And truly grateful to the V Foundation, also to the NIH Center for Reducing Cancer Health Disparities, and to all these patients. I think I might have gone overboard, but thank you so much for your time.
Thank you very much, Dr. Guda. It was great and well worth the extra few minutes. So thank you so much.